Hello everyone. Uh, Today I will be discussing about pathogenicity and infection. This is module 9, and during last two lectures I mostly discussed about virus as well as bacteria, and today I will be mostly focusing on understanding microbial pathogenicity and infection, followed by virulence factors of both bacteria as well as viruses, and finally exposure and transmission of pathogens. So here, like if you see the understanding microbial pathogenicity and infection, so there are different types of microbes. It includes bacteria, viruses, different types of fungi, and even some unicellular organism, some eukaryotes, they are also pathogen, as well as some multicellular organism, they can also be pathogen. Now, like what is pathogen and what is host, that first I will clarify. So the relationship between two organisms can be very complex. So a larger organism in this context that supports the survival and growth, particularly survival and growth of a smaller organism, is called the host. And any organism that causes disease is known as pathogen. Here I will be mostly focusing on two different types of pathogen. One is bacteria and the other one is viruses. And their ability to cause disease is called pathogenicity. So I just discuss about host, pathogen and pathogenicity, and together we say host pathogen interaction. Like pathogen is coming into host organism and then some kind of complex relation, and there are many things that actually determine whether some infection, some disease will happen or not. So here if you see the chain of infection, so here this is very interesting figure. So if I just go through this first, that the infectious disease process requires a specific agent of substantial virulence is exposed to a susceptible host in an appropriate dose. So as a result of that, if you see the chain of infection, so first one is agent. What kind of agent this is? This is some kind of, you can imagine that microbial agent, some pathogen, for example some viruses or bacteria. So this is the agent. And then if those bacteria they have, or those viruses, if they have some kind of virulence factors, some kind of virulence, that means they have the ability to cause the disease, so that is called virulence. So then this is virulence factor of virus, bacteria or any other pathogen. Then some pathogen, they need to be exposed to host organism, that's why this is exposure. Followed by another very important thing is dose, because although some pathogen they are actually virulent, so that's why they are causing disease, and we are mentioning that as pathogen. But the question here is, if the dose is very small, the number of pathogen infecting a host is very small in number, then again it will be a problem. So there should be sufficient amount of dose so that they can create the problem into the host, right? So as a result of that, dose is very important. And finally, the susceptibility of the host. So some host can be very resistant against a particular pathogen, so that although that pathogen have all these properties, but still host can defend that. And at the same time, if host is susceptible, so then the pathogen will be establishing the disease or the infectious disease inside the host. So that's why I am telling this, the chain of infection. And here, if one of this step is broken, if you just stop one of this step, then that infection will not progress. For example, if some microbes they don't have this virulence, then it is not a pathogen. It cannot cause the disease. Or I would say the dose is not sufficient. Again, it will not cause the disease, right? So that's why this is very important and interesting to understand. Now, the virulence of bacteria particularly I will discuss here, and then after some time I will be also discussing about virulence of viruses as well. Now, many bacterial virulence factors that facilitate survival are encoded in unique sequence of DNA that are readily swapped between bacteria through horizontal gene transfer. This is very important, and this is happening in bacteria particularly, I would say, in microbes, right? So, in our body this is not happening, that horizontal gene transfer. I will be discussing that. But here if you see E. coli cell, here you have this just one chromosome, E. coli chromosome. On the other hand, this bacteria, Shigella flexneri. So, here if you see, it has one plasmid and the virulence plasmid containing virulence gene. So, it will code something, and that is why it will show its virulence. So, it is pathogenic, it will cause the disease to a host. And here Salmonella enterica, this is another pathogen, and the pathogenicity islands, these are very I would say important terms in context to virulence of microbes. So, in the chromosome itself some regions are present, those are called pathogenicity island. So, that encodes multiple factors, maybe it can be proteins, so multiple factors which actually leads to I would say provide some virulence factors to that particular microbes. So, this is called pathogenicity island. Okay. But the major question here is that bacteria, some bacteria might not be virulent, they do not have the power to infect and cause the disease. But the thing is, they can acquire that power from horizontal gene transfer. That means another bacteria, like they have some virulent genes, they can acquire virulence genes from those bacteria also. But there could be different procedure I will be discussing very briefly. But this is very important in overall understanding this concept. So, the first one I would say transformation. So, this is one strategy of horizontal gene transfer. So, transformation particularly I discussed in module 1 during Griffith's experiment. So, transformation means like bacteria, they are uptaking free DNA present nearby. So, as a result of that I would say this is one bacteria and it has its own chromosome. Now, for example, in the nearby you have some DNA available, it can be plasmid also, it can be other DNA. So, better if I say this is plasmid. Okay. So, as I already explained that plasmid also contain virulence gene. So, now although initially this bacteria is avirulent, that means it cannot cause the disease. For example, during Griffith's experiment I mentioned that rough surface bacteria, that is Streptococcus pneumoniae, it cannot cause the disease, right. But whenever it took some DNA, then it becomes smooth surface bacteria and it causes the disease and it kills mouse, right. So, similarly, if they take this free DNA, then this avirulent bacteria now can be virulent. That means it can cause the disease again, it can be lethal, right. So, this is called transformation of taking free DNA, and this is happening in nature. So, as a result of that, this way bacteria can acquire this virulent factors from other bacteria, from other from the surrounding. Now, number 2, conjugation. So, conjugation is a process where bacteria, they are taking DNA directly from another bacteria, not free DNA from the surrounding. So, here what will happen, I would say, if this is one bacteria, So it has its own chromosome, but again this is avirulent. Okay, so I am just naming this bacteria as A, bacteria A. And now in the surrounding you have another bacteria. This is bacteria B, but it has some kind of virulence gene. So it encodes some virulence gene. Now, as I discussed before, that some of these bacteria, they have fimbri, they have pili, particularly they do conjugation by some kind of pili. This is called pili. Okay. So through this pili-like structure, they directly communicate between two bacteria. So this DNA can come in this bacteria, and now this avirulent bacteria, now it will become virulent. So this is another way of horizontal gene transfer. And the number 3 is transduction. Transduction is a process where bacteria acquire this DNA from through some viruses. So in this case what will happen, say for example this is one bacteria. Now this bacteria can be attacked by some virus, particularly this virus are called bacteriophage. So bacteriophage, so they will inject DNA into this bacteria, and this phage virus, they are actually attacking many bacteria at different time point, and they acquire some virulence encoding gene. So therefore they can also transmit, they can also send these virulence encoding genes to this bacteria, and now this bacteria become virulent, although before this process it was avirulent. So this is called transduction, when virus is injecting DNA into some bacteria. Okay. So as a result of that, because of this horizontal gene transfer, bacteria particularly, they are very powerful. Sometimes this is one of the reasons why they acquire antibiotic resistance. So some bacteria, they are still susceptible to some antibiotic, but over the time they will acquire some new resistance gene and they can defeat that particular antibiotic. Now I will particularly discuss about some virulence factor. So as you can see here, this is host cell, and you can see some bacteria here. I am just showing in purple color. Just imagine that these are gram-positive bacteria, that is why. So now these bacteria, they have different types of adhesion factor, which are also called adhesins, right? So which are also called adhesins. There are many adhesion molecule can be there on top of bacteria. So particularly fimbri, for example, it helps in adhesion, and also some bacteria, like as you can see here, these are the bacteria, they have some kind of slimy layer outside. This is called capsule layer. This again, this also help in attachment with host, right? And not only attachment, like sometimes this capsule, this slimy layer, they can also act as virulence factor. Like again, I am going into Griffith's experiment. As you already know that the surface, whatever the slimy layer that present, the polysaccharide layer present outside that Streptococcus pneumoniae, that prevent, I would say that like evade the host immune system. So host immune cell cannot quickly phagocytose those cells. So as a result of that, this layer, the capsule layer, is also very important like virulence factor beside its attachment. Now more specifically, if I say here I am like providing one example of Listeria monocytogenes. This is one intracellular bacteria. So this bacteria enter inside the host cell and it causes listerosis. So this is some kind of I would say foodborne pathogen, and it enters inside the host cell, and then it is causing the problem to host. So what happened? This bacteria, Listeria, so this is Listeria monocytogen. So they have some protein, this is adesin. This is called, the name of this protein is internalin A. Adesin is a group name, like some of the adesin molecule present on the surface of bacteria, we are saying those are adesin. But internalin A, this is the specific protein present on Listeria surface. And now this internalin A interact with E-cadherin. E-cadherin I discussed in some different lectures that E-cadherin, they are very prominent cell adhesion molecule present on epithelial cell. Like in our body, in our epithelial cells, lot of E-cadherin present. So as you can see, internalin directly bind to E-cadherin and that drives endocytosis. I am not going into the detailed signaling procedure, which is not that much important here. But the thing is, what I am trying to say, this specific interaction between internalin A and E-cadherin that drives endocytosis so that the bacteria can come inside the host cell. And as I already mentioned, this bacteria is intracellular bacteria. So as a result of that, now the bacteria can multiply and it can cause the problem, right? Similarly, not just Listeria, there are many bacteria. Like I am just giving one example, and another example I should give. So here, all of you are aware, that is Mycobacterium tuberculosis. So this is commonly known as MTB, Mycobacterium tuberculosis. So all of you know that it causes TB or tuberculosis disease, right? So now it is also a intracellular pathogen, right? It is also an intracellular pathogen. So now what happened? So it binds to, I would say, mannose receptor. Mannose receptors present on host cell surface. So mannose receptor, that means some mannose-like structure, some mannose-like chemical moiety is also present on the surface of this MTB. So therefore, it binds to mannose receptor present on the host cell, for example, human host cell, and therefore it leads to internalization of this bacteria. So now another virulence factor, I would say it's present, or they utilize, like they have some lipoprotein, particularly lipoprotein and lipopolysaccharide. So this Mycobacterium, they have lipoprotein and some lipopolysaccharide as their virulent factors. So now, this is present on MTB. So this is present on the surface, for example. And now in our, in host, particularly some immune cell, for example, macrophage. So macrophage, we have, again, if I say this is macrophage, so this is some immune cell, and on the surface of this macrophage, it has TLR, toll-like receptor, TLR2, TLR4. So these lipoprotein and lipopolysaccharide that binds to this TLR, and that again drives internalization of this bacteria. So as a result of that, they establish infection. Okay. Now, not only they are going inside the host cell, and after that they can use the host cell cytoskeleton for intercellular movement. So this is very, I would say, interesting aspect. As you can see, this bacteria, so it is coming into the host cell, and then this bacteria escaped from the phagosome, like this is phagosome, okay, when they are entering inside the host cell. So then this bacteria, they are escaping from the phagosome, and they are using host cytoskeletal material, for example, actin. They are actually nucleating actin, and they are making some kind of actin tail. So, this is actin tail, and they are using this actin tail to move inside the host cell. Not only that, sometimes they leave the host cell and then they again infect some surrounding cell, as you can see in this figure. So, as a result of that, you can understand how they are exploiting our own system. So, they are actually using their host actin for their own good. So, just some kind of little bit of mechanism. For example, if you consider this is Listeria monocytogenes, then this bacteria on the surface, they have this protein ACT-A. It binds to some protein, and therefore that link to actin, and therefore they establish this actin tail, and therefore they can move inside the host cell. So, now beside this intracellularization, so they are going inside the cell and they are moving inside the cell, and they are also infecting the surrounding cell. Beside that, another important strategy of bacteria is producing toxin. Different types of toxin they can produce, and they can cause problem for the host. So, as you can see here, bacterial virulence genes sometimes encode effector proteins to deliver toxins to host cells. So, this is true for many bacteria. Here we are giving some example with anthrax toxin as well as cholera toxin. So, here the idea is there are many mechanisms mentioned in this figure, but I would be mostly focusing the basic concept that here in this toxin, particularly if we consider anthrax toxin here. So, it has two subunit, A subunit and B subunit. So, B subunit here is the effector proteins. So, B subunit that binds to toxin receptor, that you can see here. This is the toxin receptor present on the host cell surface. And now this B subunit, it forms some kind of multimeric structure. So, this is the multimeric structure of B subunit. So, therefore, now it will bind to A subunit, and A subunit is actually the real toxin, but B is some kind of carrier. So, it is helping to take the toxin A inside the cell. So, as you can see here, this is coming into the cell, the host cell, and it is from the endosome, and finally, this A subunit will be released into the cytosol, and this is the toxin. This is the lethal factor, and it will cause the problem. And depending on what is the toxin and from what bacteria it came, and different types of disease might be established inside the host. For example, as you can see, in case of cholera toxin, particularly if you focus cholera toxin, then it can cause diarrhea. But the overall, if you see the entry mechanism, in this case, bacteria itself, they are not entering inside the host. This is the major point I am trying to cover. So, in this case, previously I mentioned Mycobacterium tuberculosis and Listeria, they are entering inside the host cell. But in this case, bacteria, they are producing some toxin, and they have some complicated mechanism so that the toxin can enter inside the host cell, and therefore they can create some problem and some disease symptoms. And if you see here, another interesting type of toxin, right? So, here channel forming, or you can say pore forming toxin. So, insert itself into the host cell membrane, and therefore it forms a channel or pore. So, as a result of that, as you can see, this is the host cell membrane, okay? Host cell membrane or lipid bilayer, and it is kind of continuous layer, right? Some integral proteins are there which are important for physiology. But now what happened? Some bacteria, they can make this pore forming toxin, and it will be inserted into the membrane itself. So, therefore, it will make some kind of gap here. So this is some kind of gap. So this is called exotoxin forms pore in the membrane. And therefore, what will happen? The cytoplasmic content can come out, and then some other things like some nutrients, some molecules can go in and out, and therefore what will happen? It will result in an osmolarity shift. So as water enters the host cell and the cytoplasmic content moves out. So as a result of that the cell will be lysed, right? So this is also very common. So for example, hemolysin A, this is some kind of pore forming toxin present in some pathogenic strain of E. coli, right? So I will be mentioning another interesting type of toxin, then I will move into viruses. So here, so this is again another very different category of toxin. So this is called I would say superantigen. So T cell activation by superantigen stimulates massive amount of cytokine production that result in shock and death of host, right? So this T cell activation part I will discuss in much more detail in the next module when I will be discussing about our immune system. But very briefly, if I want to say here, so what is happening? This is like antigen presentation happen for some for particularly for adaptive immune response, that the host cell, so some cell, particularly antigen presenting cell, they have here this is called MHC or major histocompatibility complex. As I already mentioned, I will be discussing in more details in next module. Now this MHC presents some kind of peptide antigen to T cell receptor. This is T cell receptor present on T cell surface, and therefore adaptive immune response initiate. Now the problem is, like some kind of superantigen, it is completely behaving in a different way. Somehow this superantigen, they stimulate this interaction. They are not the proper antigen, but they are superantigen. They somehow activate this T cell in a massive manner, and as a result of that, lot of cytokines will be produced and it will cause the problem. One of the best studied superantigen is Staphylococcus enterotoxin B. This is superantigen which exhibits superantigen activity at nanogram concentration. So as you can see, it's very toxic at nanogram concentration, right? So as a result of that, you can understand bacteria, they they have different types of, they have multiple arrays of technique and different procedure in order to establish infection inside the host cell. And now virulence of viruses. So virus, I would say they are kind of more simple compared to bacteria because they don't have so many things, as I already discussed in the last module, like in the last lecture. So mostly they are utilizing different types of host machineries for their own good. So as you can see, HIV virus and adenovirus, I'm just giving two examples here. HIV virus, they get fused with the host cell membrane. So this is membrane fusion, and therefore they are coming inside the host cell. So this is host cell and this is HIV virus. On the other hand, adenovirus, they become endocytosed inside the, so this is then endosome inside the host cell, and finally they will come out from the endosome and they will be their genomic content, particularly I would say nucleic acid, it will come out and then it will establish, it will multiply, all those things will happen. So now this membrane fusion or endocytosis, it can be it can be properly happening if some special factors are also present on the surface of viruses, right? So I'll give you few examples. For example, this HIV, human immunodeficiency virus, HIV. On the surface of this virus, it has some glycoprotein. This glycoprotein is called GP glycoprotein 120, GP 120. Okay, this is HIV virus. And now I mentioned before that GP 120 interacts with CD4 receptor present on CD4 T cell. So, for example, this is our CD4 receptor. So, as a result of that helps to go inside the host cell. And similarly, many other viruses, they also utilize different types of virulent factor. For example, if I say poliovirus. So this poliovirus, they also have some kind of they use some host receptor, right? So they use poliovirus receptor present, like the name is also poliovirus receptor, that is present on the surface of host cell. So if I say this is host cell, so they utilize receptor, this is called PVR, poliovirus receptor, right? Now, like another common virus, I would say this is measles virus. In this virus, on their surface they have some protein called hemagglutinin. So this hemagglutinin, so it will bind to host cell. This host cell surface, they express some protein. This is called Nectin-4. This is another cell adhesion molecule present in many host cell surface, and that drives the entry of this measles virus inside the host cell. So I just give few examples. So this way many virus, they enter inside the host cell and then they multiply. Now, like as I already mentioned, just having virulence factors is not enough. So there should be exposure with the host so that they can transmit and they can cause the disease, they can establish themselves. So here, like we have to understand, in order to survive, those microbes, like they are utilizing host, particularly viruses. So as a result of that, they need to go inside the host cell and they can survive there. And also, if you think that they can cause very severe disease, for example, that host is very sick, that is sometimes not good for the microbes, because if host die, then they will not survive for long. They need to find out another host soon, right? So as a result of that, there are so much complex relation between host and pathogen. And some pathogen which causes mild disease, they establish themselves better. So they can multiply, they can infect more people, more hosts quickly. So there are different types of route that some pathogen can go and establish the disease. For example, airborne transmission. So I would say some pathogen, for example, already I mentioned MTB, Mycobacterium tuberculosis. So it can be airborne. So from some MTB patient, when they are sneezing, coughing, during that time, it can it can be airborne, so that it can it can travel through air and some small droplets, and it can infect surrounding people very close by. So this is called airborne transmission. And then contact transmission, it is direct contact, like for example HIV. This is for direct contact transmission, human immunodeficiency virus, because of some sexual contact, for example, HIV can happen. Similarly, syphilis also happen because of contact transmission, right? There are many diseases like that. Now, vehicle transmission, this is I would say some pathogen, some microbes, they are present on some kind of object, for example, and that is being used by many people, and that way some disease can be carried over. For example, sometime in gym, like a lot of people, they are going there, right? And they are utilizing the same equipment present in gym. And from there also sometime some pathogen may enter, may infect, right? So this is called vehicle transmission, like they are using some kind of vehicles. It can be some inanimate object, that's all. Now, vector-borne transmission, so they utilize some vector. This is the best example, I would say, malaria and dengue. Like for example, malaria is carried by some mosquito. Similarly, dengue, although it's a viral fever, but it's carried over by some mosquito. So this is vector-borne transmission, right? So this is malaria, the disease is malaria, and it's transmitted, I would say the vector here is mosquito. Similarly, dengue, this is again another viral disease, but mosquito also act as vector in this case. And vertical transmission, it is some kind of like unique thing, not very common here. Vertical transmission means here, for example, some unborn child, like before birth, it can acquire some infection from mother itself. Although, as I'm saying, this is very uncommon, but it can happen. This is vertical transmission. This is mother to unborn child. Okay. Now, this is the last slide of this presentation, the infectious dose. So some microorganism can establish an infection with a very few in number. On the other hand, some microorganisms are required in very high number to establish infection. Okay, both are possible. So as a result of that, like there are some infectious dose which is called ID50. That means the number of microorganisms required to cause clinical disease in 50% of the inoculated host. Like it's kind of experimental setup, I would say. Similarly, the lethal dose 50 means very similar concept, but here it's lethal dose, and the previously I just mentioned infectious dose. So in this case, this value refers to the dose or number of pathogens that kills 50% of an experimental group. So here kills 50% of an experimental group of host within a specified period, right? So this is called LD50. So now host susceptibility, this was the last circle of that chain, the infection chain I mentioned at the beginning, right? But host susceptibility, it will be better, like for discussion, like during the immunology lecture, that you will get better feeling about this host susceptibility. Okay, so here I would stop, and you can follow Prescott's microbiology as well as molecular biology of the cell by Alberts et al. Thank you very much.